We'll just wait for 5 more minutes so that people can join, and then I'll start. Hi, can you hear and see my screen? Can you hear my voice? If you can confirm.
Yes, ma'am.
Okay, sure. Thank you. Okay, then I'll begin. Hi, welcome everyone. Thank you for joining. So, as you know, this is a additional problem-solving session for the course Biochemistry offered by NPTEL. And as you would have seen in your mails, so these are additional classes which are for your problem-solving sessions. So, if you have any doubt for the classes over the week, then we are here to help you with that. Now, we'll be meeting each Tuesday from 6 to 8 p.m. And the idea of these classes are that if you have any problems or if you have any questions or if you have any doubts regarding to the course or anything related to the course and exam as a whole, you could reach out to us. And for me particularly, if you ask me any question, I will be there to help you. If I can't solve it in the same day, I will definitely come back with the answer for the next week. um, and like because it is an additional session, I prefer it not to be completely theoretical, because you already have access to all the coursework material, so you already know what we are going to study in this week, the particular course, like topics for the courses. So what I prefer to do is have more of a question answer based session. So I will have a lot of questions that will help you, like revise the topics and understand the topics better. We also upload the recorded sessions for these classes. So in case you miss it, or if you're not able to understand in the live session, you can always access to the recorded videos. You will see in my presentation that I will generally ask questions and wait for 30 seconds or more like so. That's for you, for the people who are attending the live course, for them to answer. If you are attending online, you can skip that part, or you can pause and give your own answers and check with that, because all the answers will be discussed in that session. Before I start and I begin talking, I am Paulomi. I am a PMRF scholar. I am from CES IISc and I work on ecology and evolution, particularly on the molecular phylogenetics of snakes. But yes, that's my introduction. But like I said, I work on ecology, but the course about biochemistry. And feel free to ask any question related to biochemistry to me about this course. I am there to help you. Okay, so let's begin. So what I noticed in the first week of this course, the curriculum generally covered a lot of subtopics. It started with cell, and I said I will not have much theory, but I'll have some slides which give you the backdrop of what exactly happening in this week's lecture. So in this first lecture, you would have started with looking at cell. Now, cell, I'm pretty sure we are all very much aware of. Cells are of two types, prokaryotic and eukaryotic cells. The basic difference being the organization of the nuclear material. For example, in prokaryotic, you have a nucleoid, while in eukaryotic cells, you have a well-defined nuclear structure with a nuclear membrane. But the majority of the cell is covered by something called cytoplasm. And as discussed in the lectures, this cytoplasm contains central metabolites. Now, what are these molecules? What are these metabolites? They are amino acids, nucleotides, sugar, and different kinds of acids, carboxylic, monocarboxylic acids. Now, these molecules, they are all part of some or the other pathway. They're all part of some or the other receptor. They're all part of some or the other interaction. And these interaction, these pathways, as you would know by something called evolutionary developmental biology, evo, these pathways have been seen to be conserved throughout the course of evolution. So the molecules, if you know from one of the individual or one of the species or one of the animal of the animal kingdom, you can have a corollary of what the action will be in some other animal. So this is the universality of the molecules. And this entire molecules, small molecules of a cell is called the metabolome. Now, if you look at a cell, when you take the cell and you centrifuge this, what you observe is that there is a supernatant in the palate. Palate, as you would know, contains all the high weight materials. So all the particles, all the organelles, all all the heavyweight components. But the supernatant is what you will have most of the cytoplasm, where you will have enzymes, RNA, and all these metabolites, right? So more on this with the use of these questions. Because it starts with cell biology, I thought let's just put this two questions, which are also part of your, if you would have seen your zero assignment, these are part of those assignment questions. So these are just basic questions to test your current knowledge about cell biology, molecular biology. So these questions, as I will read out the question, wait for you to answer, and then I'll discuss the answers. The first question here is, who coined the term cell? And the second question is, which of the following cell organelle is not membrane bound? I am waiting for your answers. Let me know when you are typing it out. Or if you want to unmute and let yourself for to speak, that's also fine. And in case you are not able to answer, that's also fine. I understand sometimes it's not some internet issues or something, it's fine. Yes. Okay. You might have to, if you want to ask something, you might want to unmute, because I'm probably on the other screen, so I might not be able to see the screen.
Madam, the answer is Robert Wood.
Okay, and the other one?
Endoplasmic reticulum.
Okay, I will wait maybe, because I can see people are typing, so maybe I'll wait.
Ma'am, endoplasmic reticulum.
Okay, sure. Anyone else, or I can give the answer? Okay, so I will go to the answer. It's ribosomes and Robert Hooke. Robert Hooke, everyone got correct. The second one, I've got a lot of answers. So maybe as a thought for for your thought. I would let you explore the answer for this. I will give the reasoning for this maybe in the next session. Why is it not a membrane bound cell organelle, and how would you differentiate it? Okay, but from here we can just discuss whatever comes on the screen. So this one, you have 4 questions here. The first one says the entire set of small molecules in a given cell is called what? Second is dash is a secondary metabolite used to treat malaria. Very known compound. Third, many biomolecules are polymers assembled from relatively simple. What are the components that are used? And the last one, you have the small molecules inside the cell. So how are they retained inside the cell? What is trapping them inside the cell? Hmm. I see someone writing ribosome is RER bound. RER is again, if you could say what is the full form of RER? I'm asking, so if you remember the question, it said about cell membrane, which one has no cell membrane? I can go back. So it's not a membrane bound. So even if ribosome is attached to endoplasmic reticulum, we are talking here about the organelle itself. So if you look at ribosome, exactly. So you are looking at ribosome, which is connected to the endoplasmic reticulum. I'm talking about just the ribosomes, and the ribosomes as a structure doesn't have a cell membrane attached. It is actually the ordinary organelle which doesn't have this cell membrane attached to it. But if you have doubt, let me know. I will get back to you. This is what I remember and this is what I know. But if you have anything which you think is not correct, please let me know. I will definitely read up and come back to this. But yes, coming back to these 4 questions, yeah. Were anybody with the answers? So I think one of this was already in your slides, or probably 2 of them. So it's not, it's so questions are from the slides or from the related topics in today's lecture at least. Okay, someone says phospholipid bilayer, which I'm expecting for maybe last question. Someone says quinone for the second answer. Okay, wait for 1 more minute for you people to think on the answer, and then I will discuss the answer. Okay. Like I said, it's perfectly fine. I will be discussing the answer. All right. The first question says the entire set of small molecules is what I also was discussing when I was discussing here. The last point here, the entire collection of small molecules is called metabolome. Metabolome is basically all the metabolites of the cell. Metabolome, or when you say transcriptome, it means the transcripts. When you say proteome, it means the protein. So entire content of the cell is basically own, and whatever you're referring to is the first part. So metabolome is for the metabolites. So second one is quinine, not quinone. Quinine is the one that is used to treat malaria, the derivative of quinone. Third is many biomolecules are polymers, which are relatively from simple precursors. Someone wrote monomers. That's yes, but the monomers are from precursors. So the correct terminology to use is precursor, and that is what you will find in your lecture as well. And the fourth one, someone let phospholipid bilayer. That's correct, but where? Where are the phospholipid bilayer? They're in the plasma membrane. So the plasma membrane, which is surrounding the cell is what is trapping the small molecules there, right? Okay, next. So here are some points about secondary metabolites. Which of these do you think are true, is or are true? Let me read them out for you. So these secondary metabolites, they are required for growth and they offer a selective advantage. They are not required for growth and that's why they do not offer any selective advantage. These are specialized molecules that can also be toxins. And the fourth one is they are specialized molecules that can play a role in defense. So which of these points do you think are true for secondary metabolites? Second and third. Okay. First and third, first and fourth. Okay. Okay. First and fourth, first and third, second and third. Okay. Third. Okay. I'll reveal the answer and then I'll wait for you to look into the answers and see if it makes sense to you. Okay, so what I'm saying here is the true sentences out of these four options are: one, they are specialized molecules, of course they are. They play a role in defense. Someone said fourth point, correct. Someone also said third point, which is also correct. They are toxins. So if you look into secondary metabolite, they are basically those molecules that are not primarily used, but they have additional purposes. Those additional purposes are for defense and offense. So toxins and defense. Now, what happened to the first and the second point? This is what is the idea there. So secondary metabolites, which are a group of natural metabolic products, they serve as defense chemicals, quorum sensing, metabolites in environmental interactions, symbiosis, all these additional activities. Now, that's why, because these are defense chemicals, they provide selective advantage, but they, like I said, these are additional, so their absence does not compromise their vegetative growth. So what happened in the first option? It is saying that it is required for growth and conferred. So the second part is correct. Conferred a selective advantage, yes, that's correct. But required for growth is not correct. It is not required for growth. Uh, Organism can do without secondary metabolite. The second option says it is not required for growth, which is correct, but it does not offer a selective advantage. That is wrong, because if you have secondary metabolite, if you have the defensive material, if you have those extra materials which help you in quorum sensing, symbiosis, then you definitely are at an advantage compared to your competitors and compared to your species. So that's why the first and second are wrong. The third and fourth are correct. If you have any doubt, please let me know. Uh, uh, Second and third is what I said, right? I hope someone wrote second and third. I hope that makes sense to you. Which metabolite plays vital? Which metabolites? So all the byproducts of the pathways. You have several pathways in action, right? TCA, Krebs cycle. So all the metabolite, all the components that are used on those in those pathways, those are the primary metabolites. Those are also synthesized from, like I said, from precursor molecule, which are also fed upon by, like these are also made by several proteins and several amino acids, several uh, carbohydrates. We'll see. So all of these together made up metabolites, which are used in these important pathways. Those are the primary metabolites. The secondary metabolites, these are also part of these pathways only, but they are as extra. These are the secondary byproduct, which are then used for other purposes. So if you see quinine, someone wrote here quinone. Quinone's derivative is quinine, and this quinine is what is allowing the, so we treat malaria, but that's because what we repurpose it to use. But quinine in the animal is used as a defensive material. These secondary metabolites are most widely studied in plant, if you know, and if you see in plants, they have a whole repertoire of secondary metabolites that are used for defense materials. So you see that those metabolites that they are used from, the precursor will be part of a primary metabolite. Yeah. So I hope this made sense to Varun. And I see that the person who said then second and third says okay. If no more doubt, I'll move on to the next question. Okay. All right. Right. So, like I said, the like I saw in this three lectures, they're basically compartmentalized into several subparts. So, the first part was just a general introduction to cells. Second part was about macromolecules, about the components of the cells. So, what we looked into was polymers of these different biomolecules, and like I mentioned, they are assembled from precursors, which after forming are further assembled into supramolecular components so that they form the functional unit. So we have four kind of macromolecules: lipids, nucleic acids, carbohydrates, and proteins. So on your slide right now, on the left side, you see carbohydrates, which have monomeric and polymeric units. Proteins, which are in their linear form, it's the correction, sorry, like linearly an amino acid being joined, that's a primary structure. Them folding uh, their folding gives rise to different kind of suprastructures. So secondary structure, tertiary, and quaternary. Quaternary and tertiary are important for their functionality. Nucleic acids, we have DNA, RNA that consist or that made part for the genetic material. Uh, if you look at nucleic acid, DNA, or RNA, the basic components are bases, nitrogenous bases, a sugar, a pentose sugar, and phosphate group. Now, the connection between nitrogenous bases and the sugar is called nucleoside. When it is these two units are joined, they are nucleoside. When on nucleoside you add phosphate, it's called the nucleotide. And this nucleotide as a whole is part of the DNA or RNA. And lastly, you have lipid. So lipid is basically, as you can see here, you have it basically is found in these polar components. So it's basically facing away from the water. So it forms, if someone wrote phospholipid bilayer, so it's part of the lipid will be inside because it is facing away from the water, and they have different type of lipids which form part of different components. What I mean, it's something that you will see in further slides. Right, coming to questions before we discuss any further by text. These questions, which of these polymers are not naturally found within any kind of cells? And next, which of these letters are used to signify the four nucleotides found in DNA? Easy question. This shouldn't be at all difficult.
Polyethylene.
Yes. And for the second, which is, I think, the easiest question. Yeah. As I see, almost everyone who's on the call has got it right. So that's correct. Let me just switch to the next screen. Polyethylene and ATGC. Polyethylene is something that is not at all natural, it's a synthetic material. And ATGC is adenine, thymine, guanosine, and cytosine. They got it right. Quickly moving to the next one. So which molecule is involved in the transfer of genetic information? First, second question, most of the enzymes that you see, they are which component, like they are which biomolecule? And the third one, what is common to DNA and RNA? See the question properly and then you answer. What is common to DNA and RNA? Okay.
First one is nucleic acid.
Okay.
Enzymes are proteins.
Okay. And the third one?
Phosphate backbone.
Just a second. Phosphate backbone. Okay. Anyone wants to add to these answers for any of the questions? Phosphate backbone, protein, sugar, phosphate, nucleic acids. I see a lot of nucleic acids as the answers. I see some sugars.
Complementary base pairing between guanine and cytosine.
Yeah. Okay. I see some people typing. Should I wait for maybe? Okay. Wait just a second. All right. Let me give the answers. If people are still writing, you can check. All right. So first and second, most of you, I think all of you got it right. The first is nucleic acid, second is protein. That's correct. Third one, people wrote sugar, if I'm not right. So let me, if I'm not wrong. So let me just tell you why these two are not correct. So the correct answer for, or the common components between DNA and RNA is phosphate backbone. That's correct. Second, GC. GC is present in RNA. You don't have T, but you have G and C. So GC pairing is common in DNA and RNA. Now, sugar, what is DNA's full form? Deoxyribonucleic acid. What is RNA? Ribonucleic acid. So ribose and deoxyribose are two different types of sugar. That's why they are not same component. And base composition, DNA has ATGC while RNA has AUGC. So these are not similar. So I hope that made sense because I saw some people writing sugar. So I hope that makes sense. I'm taking the silence as a proxy for everything being fine. Just a second. Just forgive me. All right. I'm so sorry. Moving to on. Dash is a long unbranched polymer of glucose, while dash is a branched polymer of glucose. So basically, which one is branched and which one is unbranched? What do you think is the answer? But if you consider eukaryotic mRNA, G and C pairing would not be. Okay. I'm sorry, what? So I am not talking about standing in ends. When it is single stranded, we are not even talking about complementary base pairing anyway. But when mRNA has to bound to the, you remember if the tRNA and the mRNA, the pairing will always be between G and C. That is what we meant. So A and T pairing and G and C pairing will always be, just a second. Sorry. So A and T or A and U will always pair and G and C will always pair. It will never be G and A or A and C. That is what I meant when I asked those questions between the common point. So the pairs that will be formed, that will always be constant between for RNA and DNA.
Amylose and amylopectin.
Okay. Sure. Yeah. Anyone else? Amylose, amylopectin. Second option. All right. Yep. You people are correct. And this is a summary of all these different subunits and their branching pattern for the three component of polymers of glucose. So cellulose, glycogen, and starch. So cellulose have beta glucose. Beta alpha again is based on their structure of the glucose when you look into them. And they don't have branches because they are just, so you can see the shape of cellulose and the shape of starch. In starch, it has two components, amylose and amylopectin. As people have correctly guessed, amylose is the non-branched or unbranched polymer, while amylopectin is the branched polymer. For glycogen, it uses alpha glucose and it has again a 1, 4, and 1, 6 pattern branchings. So this is a summarized version of the other kind of polymers that are there. Okay, next. Dash bond is between two monosaccharides. So when two monosaccharides are joined, what kind of bond is formed? When two amino acids are joined, what kind of bond is formed there? And the next question is, if you look into cellular membrane, what is it typically made of? Again, very easy questions. But because we are revising, I we have put these all questions here.
No, the first one is glycosidic linkage. The second one is peptide bond.
Yes. Okay. Thank you. I will maybe wait for the third question answer and then I will. Cellular membrane is made of phospholipid. I see some people typing. Okay. And yes, anyone? Yes. So I see everyone is very well aware of the answers, and all of you have got it right. So monosaccharides, if they are forming a bond, that's glycosidic bond. Amino acid form a peptide bond, and cellular membranes are form of phospholipids. That's great. Okay. So we extract DNA from a given sample, and then we do absorbance ratio test. We look into the absorbance ratio. So which one of these options do we look for or we use to check the purity of DNA sample? Okay. Can anyone also tell me what should be the ideal purity? So I see options of 260 by 280. So I do 260 by 280 test. What should be the ideal value? Okay, correct. Someone has written 1.8. All right. Now I will give you one additional question. Among these options, there is another set of absorbance that we look for as a secondary test to DNA purity. Can you guess that or can you think about that? Yeah, range is 1.72. Correct. That's the ideal range. The ideal value is 1.8, but the range is correct. 1.6 to 2 basically goes as an RNA contamination. So 1.6 to 1.8. But other than the answer that you have all written, 260 by 280, there is one more value that pops up when you are looking into the purity. Could you guess that? Or do you know that out of the given option? Okay, I will give the answer. And for people who are still wondering, no worries. I will also give the reasoning. So 260 by 280, everyone has written. And if you're not thinking, this is the correct answer for that. And 1.8, as I have already discussed, 1.8 is the ideal value. The range is somewhere around 1.6 to 1.8. If you look into a value of 2, we generally take it as an RNA contamination. And visibly, so if you put RNAs while your experimentation, you will definitely see a drop in the call it absorbance from 2 to 1.8 and 1.9. The other one is 260 by 230 ratio. So this is again taken as a secondary measure. And here, in this case, the ideal ratio is 2 to 2.2. There now, if you have a lower absorbance here, that's taken as again additional contamination. So we have contaminations, for example, proteins or some other kind of cell debris that not debris, but some other kind of cellular metabolite that can come into your DNA allure. If those are present and they have at higher quantity, then this 230 nanometer absorbance for those will be lower, and that will give you an idea about your purity of your sample. So the first one is A260 by 280. We also tend to look at 260 by 230 for a range of 2 to 2.2. Did that make sense? Any doubt? Okay, moving on. Now we have 5 statements on nucleic acids. Which of these are incorrect or false? So the DNA backbone is composed of phosphodiester linkages. The two strands of DNA run antiparallel to one another. Adenine and thymines are purines. Nucleosides contain nitrogenous base, pentose sugar, and phosphate. And uracil is present in place of adenine. These are not difficult, but take a time to read the statements properly and then attempt to answer.
Adenine and thymine are purines.
Okay, I'm assuming you're saying that's one false answer. Okay. 3/4, 1/4, 3/4, 5th. Let me look in the answers in the meanwhile. Please be mindful you are writing about false statements, okay? The question is false about the false statement. So I hope you're taking care of that. Right. 4. Okay. 3, 4, 5, 3, 4, 5. Okay, I see people have written 3, 4, 1, 4, 3, 4, 5, 5, 3, 4, 5, 1, 3, 4. Okay. 3, 4, 5. Okay, I see no more typing, so I will discuss the answers. 3, 4, 5, as most of you got it right. I saw some 1 or 2 answer on 1 also. So let me tell the answer here. The first statement about DNA backbone being composed of phosphodiester linkages is correct because you see this here, that nitrogenous bases, sugar, and phosphate. This phosphate is what links the different bases to each other. So DNTPs, all the nucleotide triphosphates, are linked to each other by phosphodiester linkages, and that forms the backbone of the strand of the DNA. So you have two strands running with along parallel to each other, and they are bound to each other by the hydrogen bonds. But the strand is held together, the backbone is held together by phosphodiester linkages. So that's why this first and two are correct. Third, fourth, fifth, as most of you have guessed, is correct. That the others are incorrectly stated. Uh, Anyone? Yes? No? Okay. I'll come back to keep, if like I said, please put your doubts in the chat box. I'll keep coming to the chat box and keep checking it. All right. The third one says adenine and thymine are purines, and on the right of your screen, you can see what are purines and what are guanine are purine, thymine and cytosine are pyrimidine. These are for DNA. Now the fifth point, uracil is in place of thymine. Thymine is called 5-methyl uracil here. Rest everything is same. So thymine so that's why uracil is present as a substitute for or in place of thymine. And the fourth one, which I have already shown, nucleoside is basically base and the sugar. When you add a phosphate to it, it becomes a nucleotide. Can you explain about determination of DNA sample of and absorbance ratio? Yeah, sure. So basically, when you are looking into the purity of DNA which you have extracted from a sample, what you do is you use different kind of method, for example, nanodrop method or spectrophotometer method, where you're basically trying to look into the absorbance of the DNA at a certain wavelength. For our reference here, we look into the 260, 280 wavelength, and then we look at the ratio of these two absorbance. So we look into 260 by 280, these nanometer wavelength ratio, and we look into the purity. Now, as someone mentioned in the chat, the purity range is from 1.6 to 1.8. Why is it important to look into the purity of your DNA? Because it gives an idea of what you have extracted. Have your DNA extraction process gone right? If it's 1.6 to 1.8, by my experience, I can tell you that it works very good for PCR. But if your subsequent steps are, let's say, cloning, or let's say you have something which involves very precise DNA concentration and purity, then you should always target towards 1.7, 1.8. But if it's only PCR and just looking into the, for example, like I work on species phylogenetics, so I just want to sequence a particular marker. For that PCR, 1.6 works fine. Now these are this is the primary thing that I look into when I do DNA extraction. But I also look into 260 and 230 to have an idea about my contamination. What contamination? Protein contamination. Let's say some lipid contamination that would have been carried over during my DNA extraction. Hello? So why is that important? I could only look into DNA and be okay with it. Why should I look into this other absorbance? It's because these proteins, these carryover contaminants will cause a problem during my subsequent procedure. Now, I want to do PCR, but sometimes these proteins can cause a hindrance to my PCR. I want to do cloning, but having more of contaminants just diminishes my chances of enhancing the quality of DNA in the further steps. So that's why when you are looking into the absorbance of the DNA that you have extracted, you look into these both ratios and take a call of how good your DNA is. The other check is gel electrophoresis. If you know, you also look your DNA quality on the gel. You run it on an agarose gel and see how well your quality looks like. So these are all different checkpoints of the result of your DNA extraction method. Did that make
Yes, ma'am.
sense?
Yes, ma'am. Understood, ma'am.
Okay, sure. Thank you. Um, yes. So this answer is okay. Like, did this question make sense? Anyone has any doubt? I am assuming no. So I'll move ahead. Right. So now we have spoken about metabolites, we have spoken about nucleic acids. Now let's talk about carbohydrates. We also talked. So in carbohydrates, again, you have these 5 sentences. Now you are supposed to tell me which one of these are true. So the first one is maltose is formed by alpha-1,4-glycosidic linkage between two glucose molecules. Glycogen has a structural role in bacteria and eukaryotes. Glucose has a 6 carbon molecules and is a keto sugar. D-glucose and D-mannose are epimers. Reducing sugars are capable of reducing ferric or cupric ions. Which one of these are true? 145.
One four, ma'am.
14, okay. And
1 4, ma'am.
14, okay. 124, 14, 145. Okay. Maybe I will go to the answers. So it's 145. I will wait for some people who had, I think, answered 2. Yeah, you see this, and then I will tell you why the answers are correct or wrong. All right.
Ma'am, can you
So,
explain the final option, ma'am?
sorry.
Can you explain the final option, ma'am? Reducing
Yes,
sugars are
I will do.
capable of.
Yeah, yeah, I'll do. I'll explain all the. I just wanted you to see this thing. Right. So the first statement, as you can see, when you look into the maltose structure, you will see that maltose is formed of D-glucose molecules, and these are joined by alpha-1,4-glycosidic linkages. So that's the correct answer. The next correct answer is basically D-glucose and D-mannose are epimers. So I will give this first. I will show you what are epimers and then come back to it. So epimers are basically same component, like they have the same formula C6H12O6, something like this, but they differ only at one position. Now, let's say D mannose here and D glucose here. Why did we say it's an epimer? Look at the structure. Where are they differing? They're only differing at the second position in the orientation of the H and the OH. So when you say D-glucose and D-mannose, they are epimers of each other at the second C2, second carbon. While if you look into glucose and galactose, see properly the structure and you will see that they are only differing at the fourth carbon. So they are called C4 epimers. So epimers are basically who have the same formula, structural formula, but they differ in their structure at one position, or positions where they differ are certain positions where they are differing is where they are epimers form. And next one says, the last one says reducing sugar are capable of reducing ferric. So ferric, cupric are basically Fe3 plus or Cu3 plus ions. And these sugars, they're able to reduce the Cu3 plus or Fe3 plus to Fe2 plus and Cu2 plus. And they will generally do this in an alkaline solution. So that's why reducing sugar, you will mostly see them in an alkaline solution. Reducing is basically in chemistry, so you will see, right? You reduce the oxidation state of copper or iron, ferrous ions. So that's reduction that we are talking about here. And here, in case of an alkaline solution, if this is mediated by a sugar, then it is a reducing sugar. And what will that do by reducing? It will form an aldehyde or a ketone. So mostly you will see that aldose sugar or ketose sugar, these are the type of the reducing sugars that you found. And these are mostly working on such 3 plus oxidation state ions. Okay, now I will quickly say about the second and third, and if you have any other doubt, please let me know. So the second one says glycogen has a structural bacteria and eukaryotes. I think someone wrote about second also. Structure and role in bacteria and eukaryotes are performed by carbohydrate, but not by glycogen. It is by glycan, which is on your right side of the screen. This is what you will find in the structural role. Structure basically membrane, plasma membrane. What is glycogen? Glycogen is something that you will also see in the pathways. If you see the glucose pathway, if you remember the phosphofructokinase pathway, you will see glycogen is a stored form of glucose. So it is used for metabolism. In metabolism, you use glycogen. But if you are talking about structure, you in bacteria and eukaryotes, you find glycans, not glycogen. And the third one is glucose is 6 carbon. That is correct, but it is not a ketone sugar. You can see in this part here, it is an aldose sugar. So it has a CHO group, while if you look into fructose, it has a CO group in its structure. A CO is basically keto, and a CHO is basically your aldo. So glucose is an aldose sugar, and fructose is a ketose sugar. Ketose is basically they have a keto group, and aldo is basically they have a CHO group, aldehyde. Oh, I'm so
Madam,
sorry.
Madam,
Cu2 plus
final
to Cu plus. Sorry, I am not able to hear.
Madam, final
Hello.
option not getting clear, madam.
Final not getting clear in what sense? Like what exactly are you confused at?
That is the reducing sugars are capable of reducing ferric to cupric ions, no?
Ferric
That option.
2, I don't think it says ferric 2. It says ferric or cupric ions.
Ah, yes, ma'am.
So,
Yes, ma'am.
so what is not clear in this? Basically, you're reducing the cupric ions from an oxidation state of Cu2 plus. I'm extremely sorry for the mistake. Thank you for finding that out. Cu2 plus to a Cu plus state. So these sugars help in reducing the oxidation state of the copper ions there from a 2 plus state to a Cu plus state. The valency of the copper changes. I hope.
Hmm.
Okay, if it is not clear, I will definitely put this up in the next session. Will that be fine by you? I will explain it in a proper way that way. I will have slides and I can show you if you need. If it's
Yes,
very
ma'am.
May I
unclear
explain
to
it
you. Sure.
if you let me?
Sorry.
May I explain it if you let me explain?
You can please go ahead. So it's a
Yes,
discussion
yeah.
anyways. Please
So,
go ahead.
yeah. So the reducing sugars actually have uh, the carbonyl groups free. Okay, either it can be, I mean, converted into the single OH. In that state, also it can reduce the highest oxidation state of the I mean copper or the iron. Okay, so if it is reducing cupric ions, that means Cu2 plus, it will form Q plus ions. Okay, so if suppose the molecule is cupric oxide, so it will form Q plus oxide, and it will get precipitated, which is brick red in color. So aldehyde and ketones have the reducing property actually. But if it is free, then only it can reduce that kind of salts. Or that kind of ions.
Yeah, thank you so much, Priyobrato. Thank you so much. Yeah, I hope that made it clear what he was trying to refer to. And he was absolutely correct. Let me tell you that he was absolutely correct in what he was saying. If it is still a bit of not visualizable, I will definitely put this in slides and show you by what exactly Priyobrato has mentioned today about brick red color and how they are reducing it. I will put it in a pictorial form so that it gets a bit more clearer if I couldn't be more clear. But thank you so much, Priyobrato. That was really, really helpful. Okay. But any more questions in this slide other than the last point? That last point I have noted. I will definitely include this in the next session. Okay. Okay. Let's move to the next question then. Okay. So now we are now looking into proteins and amino acids. So out of these questions, statements, which one do you think is or are false? Selenocysteine is derived from cysteine and contains selenium in place of sulfur. When dissolved in water, amino acids exist as Zwitter ions. In peptide bond formation, the amino group acts as an electrophile. Peptide bonds are highly stable as a result of high activation energies for bond formation. And last one being the secondary structure of protein referred to stable arrangement of amino acid sequences. Okay, I see people are still thinking. So yeah, please take your time.
Second and fifth.
Ma'am, second
Second,
and fourth.
fourth, second, and fifth. We are talking about false statements. I'm once again highlighting and reminding you that we're talking about false statements. So second and fourth and second and fifth. Okay.
Even fourth also, ma'am.
Okay. I'll just check. Second and third. Okay, I see people are typing. I'll wait for those answers and then go ahead. Second and fifth. One, three, five. Okay. 1, 3, 5.
Second, fourth, and fifth, ma'am.
Okay, I will, because it's a mixed lot of answers, I will first show the answer and then I will see how many got third correct. 3, 3, 3. Okay. Okay. There are a lot of confusion. I'll start with the most interesting one. Uh, what is Zwitter ion? Does anyone know? The second point, Zwitter ion. Does anyone know what Zwitter ion is?
It contains equal amounts of plus and minus. The carboxyl group can able to donate a proton, and also the amino group can able to accept a proton. So on that we get Zwitterian form.
So it is in Zwitter ion form, right? It is existing as Zwitter ions, so that's not a false statement. I saw a lot of people writing yes, it is, but it is still a Zwitter ion because your O is negative and your NH3 still is plus. So you still have those two charges. Like the Zwitter ion, by its definition, says a Zwitter ion has a molecule which is net zero charge, but it still has positively and negatively charged functional groups. And in water, when you put amino acids like someone mentioned, the carbonyl group O becomes negative and the amino acids group become positively charged. So you have both the charges on them. So second is true. I saw a lot of answers with second as the false statement. Does that make sense? Neutral charge is definitely there, but the individual functional groups retain the positive and negative charge. And they are the definition of Zuger ions. And amino acid is the canonical or the most obvious example of Zwitter ions. So, yes, if it is still doubt, let me know. Because this is important for you to understand a lot of things later. But Zwitter ions has a neutral charge because, as someone mentioned, equal amount of positive, equal amount of negative. But positive and negative both are present. Yes, correct. So second is true. Everyone is convinced. Chat box is open. Do let me know if you are not okay and you're not agreeing with me yet. Okay. First one, I think someone wrote first as well. First one is also correct because only the third one is, as I will explain in a while. The first one is selenocysteine. It is analog of cysteine and it contains selenium in place of sulfur. It is the 21st amino acid actually. Cysteine consists of sulfur SH group, while selenocysteine is basically S being replaced by selenium. So this is also true. Many people wrote fourth. Uh, Peptide bonds form because of, as you will see here, because of a very strong nucleophilic and electrophilic interaction. So it has very high activation energy. And anything which is very high activation energy makes it a very stable bond. It is not very easily breakable. It's not a weak interaction. And the fifth one, which also some people wrote wrong, which is actually correct because I had mentioned primary structure is the linear structure of amino acid. One amino acid back to back to each other is a linear structure. And linear structure is basically when you've opened up everything. Now, when the amino acid chains start folding upon itself, that gives rise to all these different structure. And as and when these chains start folding upon itself, they have secondary interaction with each other, which makes it very stable. And that's why your secondary structure is a stable arrangement. Then your tertiary is more stable, and quaternary is where you require them multi-unit to come together to function as a single unit. So first, let me know if these four made sense. Okay, correct. So Priyabrata has already also written that selenocysteine is found in glutathione peroxidase. UGA, despite stop codon, codes for it, and that's why it is present. It's also that's why it's called 21st amino acid because it's also coded for. So that is not incorrect statement. The only incorrect statement here is the third one, which is in a peptide one formation, the amino acid group is actually a nucleophilic nucleophile, while the carboxylic group is acting as an electrophile. So the statement formation was wrong here. As you can see in this diagram here, during the peptide bond formation, you have the nucleophilic attack by the amino group of the amino acid, which is here. This is attacking the electrophile carbonyl carbon of the carboxyl group, and this leads to the dehydration, removal of H2O, and this forms the CONH peptide bond, which is a very, very stable bond because it has very high activation energy. Can you please explain the fifth option? Fifth option? Okay. Maybe I'll go back to the slide quickly. Wait a minute. Give me 1 minute. Okay, slide is visible on the protein and carbohydrate slide.
Yes, yes.
Yes. So here you see proteins have four different structure: primary structure, secondary structure, tertiary, and quaternary. Primary structure is this chain here. Just consider these different colors to be different amino acid. Eight, like you can go glycine and valine and all these different. Now, two sheets, two chains coming together as helices. So you have one helix, one chain forming like this of these peptides, the other chain forming like this as helices, just one around each other is called alpha helix, right? So now you have two chains, two amino acid single chains coming and interacting with each other. You can simply imagine as a thread. If you have a single thread, single wire of thread, it is definitely weaker than if you twist it with two different strands. The moment you twist your strand, they become more strong. That's alpha helix. Now, beta sheet is basically one chain is running like this, and the other is running anti-parallel, and they form bond among each other. That gives this pleated kind of uh, structure. That is also a secondary structure because you have just one layer on top of the other. That is beta sheet. So alpha helix and beta sheet are the two type of secondary structure, which is basically the chains, two chains coming together. Now alpha helix and beta sheet forming structure together, as you can see here. If you see the tertiary structure, you see alpha helices are there, and these alpha helices are connected to each other in a beta sheet manner. That's a tertiary structure. And quaternary structure, if you know hemoglobin, if you know myoglobin, you will know that they have tetramers, they have 4 units. So all these this tertiary structure is 1 unit. Four tertiary structure coming together, it's called a quaternary structure. That's important because for hemoglobin or myoglobin to function, they need those 4 units together. But individually, those units are formed by primary, secondary, and tertiary. And at each level, after the first chain, your stability keeps on increasing. And that's why you're increasing the structural strength, because you want the chains to retain in a better and a stronger way. So secondary structure is where it's a stable arrangement as compared to the primary structure. Let me know if that makes sense. I will go back to the question again. Yeah. So the secondary structure of protein, which is alpha helix and beta sheet, is basically stable arrangement of amino acid sequence. Is basically amino acid chain that was formed as a primary structure. Does that make sense? Okay. Right. Moving ahead. The last macromolecules, lipids and fatty acids. Next, this 5 set of questions. So you have membrane lipids. They are polar in nature. Double bonds in naturally occurring unsaturated fatty acids are generally trans. So trans configuration in naturally unsaturated fatty acids. Primary carbon chain in fatty acid typically contains an even number of carbon atoms. TAG or triacyl glycerols are the simplest lipids composed of 3 fatty acids linked to a glycerol molecule. The name of the compound suggests the structural composition. And lastly, sphingolipids are present on the cell surface and plays a biological role or role in biological recognition. Which one do you think are true?
Fourth option, ma'am.
Fourth, okay. Anyone has, okay, 542, fourth, okay. Any more answers? 4th.
1 4.
1 and 4. So 1, 4, 5, 4, 2, 2, 4. Okay. Okay. Maybe I will give you the answers. It's 1, 3, 4. I'll go back and check whole total which are 5, 4, 2. Okay. 5 and 4, 2, 4th, 5th, 4th and 5th. Okay. Okay. Let me also show this. So membrane lipids, if you remember my structure, when I showed about the phospholipid, I showed you membrane has hydrophobic chains towards the inside of the cell, while it has a hydrophilic component towards the membrane, which is the phospholipid. The phosphor groups are outside. So that's why they are polar in nature. Double bonds in naturally, this is a information that you should retain. The double bonds which are found in the naturally unsaturated fatty acid, they are always in cis configuration. What is unsaturated? Unsaturated fatty acid means having a double bond. Saturated fatty acids have all single bonds. Okay. The primary carbon in fatty acid contain even number of carbon atoms. That is true. So if you generally will see linoleic acid, oleic acid all being even numbered carbon atoms. TAG, as I said, the name suggests it is triacyl. So to a glycerol backbone, you have three fatty acids. Acyl is basically fatty acids. So three acyl groups to the glycerols. That's correct. Now, sphingolipids are present on the cell surface and recognition. Well, sphingolipids are mostly present in your brain cells. There, it is mostly, if you will see sphingomyelin, it's mostly used for the insulation of those nerves there, the brain cells. So it's not the true lipid which is taking part in the biological recognition, which are basically the phospholipids and the glycerololipids, which you will see as if you look into the example of lipids, the glycerololipids, which are glycerol groups, those are the ones that are found in your cell surface. And when we talk about receptors, GPCRs and these, those are the kind of lipids that are being talked about. When you say sphingol, you are referring to brain cells, and the primary work of those lipids there is insulation, providing, if you do myelin sheath, nerve protection. So you find sphingolipids in those kind of cells. Uh, Okay, if you have no doubt, let me move on to the next part. And lastly, again, all of these questions, all of these points are a revision for you for all the topics that were studied in this week. So these are additional points, but then these are also very important. Lastly, biomolecules, so which are false for these options. Biomolecules, the ones which are mostly naturally occurring amino acids, they are found in the L configuration. And all amino acids are chiral in nature. Three important terms here. Cell membranes are composed of phospholipid bilayer. Purines and pyrimidines form the backbone of the DNA, and amylose is a polysaccharide composed of glucose unit. Read carefully and then attempt to answer. There are some important terms here like configuration, L configuration, chirality, backbone. So note those words and then try to answer. And that is what is important when you are also answering for your exams. Look into the key terms here first. Second and fourth, okay.
Second and fifth.
Second, fifth. I also have an answer on second and fourth. Others are also writing, I believe. So let me just wait for the answer. 5, 4, 3. Okay, let me give the answers. False, okay? These are the only options which are false. Okay, let me show you. Right. So L configuration, reconfiguration, we'll do it in the next slide. So I'll keep it for there. But mostly amino acids that you see in nature, all the amino acids that we know, they are mostly in L configuration. What are the L and D? I will come to them in the next slide. Important terms. No. Second is all amino acids are chiral in nature. Chirality is basically when you have 4 different groups attached to the central atom. In case of amino acid, the central atom is carbon. So when you have 4 different groups attached on carbon, that's chiral carbon. Okay. But the statement says is all amino acids are chiral. That is wrong because you have glycine. The simplest amino acid has 2H, 2 hydrogen. So when you have 2 hydrogen, that means you don't have 4 different molecules. You have one carboxyl, you have one ammonium, but you have 2 hydrogen. So you are not chiral anymore. You are achiral carbon. So glycine is achiral, and that's why all amino acids are not chiral in nature. Third, everyone got right, I guess. Cell membranes are, of course, composed of phospholipid bilayer. Fourth, purine and pyrimidines are not the backbone of DNA. The backbone of DNA is the phosphate, sugar and phosphate connection, right? Because what happens in purines and pyrimidines, they are forming the nucleotide, but their connection is by the H bond. That is not the backbone. The backbone, as I said, phosphate is what is linking the sugars with each other. And lastly, amylose is a polysaccharide of glucose subunit. This glucose unit, this we have already seen in structural when we look into the structure of glucose, starch, and cellulose, all of these. Because not only phospholipid are in plasma membrane. Wait, which one is? So in the statement, like I mentioned, when you are reading a statement, be very, very careful. The statement says cell membranes are composed of a phospholipid bilayer. It did not say only, it did not say majorly, it says phospholipid bilayer, which is the correct answer, because if you know about cell membrane, the accepted hypothesis or the accepted mode of structure that is accepted for cell membrane is the phospholipid bilayer model. So that is a statement, that is what is referred here. It did not say only phospholipid doesn't have glycerol lipids. That's not correct. It did not say it is majority of phospholipid. That's also not correct. It is saying a phospholipid bilayer, which is correct because that is the accepted model and that is the structure of cell membrane. I understand the confusion and it's okay. But next time, whenever in any exam you're looking into the answers, just be careful and mindful of the words there. Okay. Okay. Quickly. Right. Now, I said about the L and D configuration. We'll come to those configuration now. So the next very important topic of the first week was stereochemistry and stereoisomers. So stereochemistry is basically telling you how are the different atoms or the constituents of a molecule arranged in a 3D space. You can have Bostic model, you can have linear, just, like projection and showing the structure. So you have these different ways of showing how the molecules are spaced and arranged with respect to each other. And this is very important for visual interpretation and to understand how are their positioning affected. So generally, we just use something called cis, right? So cis, whenever you use this term cis, you mean same side. So if you look at the structure of malic acid or malate here, the hydrogen group or the carboxylic group or COH group, they're on the same side of double bond. They're on the same side of the overall structure. So it's cis. But now compare it with fumaric acid, they are on the opposite side. They are on the diagonally opposite side. That's trans. So the basic difference between cis and trans is cis is towards the same side and trans is towards the opposite side. Now, on the left, you see chirality and achiral molecules. This is what I've just explained. Chiral being all different molecules, but chirality also has another explanation to it. You see here, chirality means rotated molecule cannot be superimposed on its mirror image. So when you have a chiral image, chiral molecule, if you rotate it against a mirror, the images cannot be superimposed on each other, right? And achiral, if you rotate it across a mirror, the images are superimposable on each other. Look at your hands, left and right. Tell me this answer. I'll come back to chat box for you to see if you've understood. You have left and right hand. Do you think these both are superimposable on each other? If you get this concept here right, you will get all the answers correct. I've got one answer saying no. What do others think? Are your left and right hand superimposable? I'm not putting it one on top of it. I'm putting it like this, the same direction. Superimposable will mean that I can put on top of each other in the same exact manner. Okay. So, like someone mentioned, the answer is no. This is your left hand, this is your right hand, put on top of each other. Your fingers don't exactly overlap each other. So this is not superimposable on each other. And these are type of references I was just giving in the slide. So if you have two molecules and you lay on top of each other, if they perfectly align with each other, they are superimposable. If they don't, that's non-superimposable, right? So now if you come to these structures here, look into the first two blocks here. Some arrangement, CFC, H, CH, two methyl and next and Y group. 1 second. You, I am, I thought you wanted to say something, but okay. Anyways, let's go. All right, so look at the first and the second structure. You have two carbon, two hydrogen. Here also you have two hydrogen. You have X and Y. You have X and Y. CH3 on at top and bottom. Now imagine you put a this thing mirror here. If you don't want to imagine that vividly, just imagine this is a paper and you are now folding the paper on top of each other. If you fold the paper on top of each other, if you bring the right onto the left, do you think the images or the structure will be completely overlapping each other? In the first and the second image, if I bring the left side or right side of the paper onto the other side, do you think both the images will overlay each other, will be superimposable? Okay, yes. This first, let me bring the pointer. This structure and this structure. Now imagine this to be a plane of division and I am bringing the right side, taking it, picking it and dropping it over the left side paper. So do you think the structure will completely overlay each other? Okay. Okay, someone says non-superimposable, someone says yes. What I meant was, this is the sheet of paper. On my left, I have some figure. I have on the right side a figure. I just will divide this like this and make it one single unit. So when I do this, do these structures look similar? I don't mean to make and sit it on top of. I'm just folding this paper on top of each other. If I do this and the structures are on top of each other, will they look similar? Okay. Keep this, whatever paper I have shown, in mind. Okay. They are superimposable. So if what I just did, if I show you again, now see my hands, okay? They're on the other side. So this side and this side. When I fold, my hands come on top of each other. This is superimposability, right? So when you do that for these two structure, when you fold this right over the top, your X and X will match, your Y and Y will match, your H and H will match, CH to CH are always matching. These are mirror images. These are called enantiomers. Please let me know if this is clear. If it is not, I am ready to spend more time here. It's fine. I'll take more questions in the next session. But this is important for you to understand. Okay. Thank you. Enantiomers are non-superimposable. You don't think about that right now. What I asked about superimposability in my term was what I explained. Was that clear first? When I explained about the superimposition on each other? Anyone? Let me know if you have any doubt. Yeah. So now, first of all, forget about enantiomers. I will come to that later. But if you are able to do a mirror image of each other, where you look into each other clearly, if when you are matching it, this is what I meant by mirror image. Now, enantiomers are mirror images. Now, if you understood my hand example, let's now take the hand example. Remember when I said hand are non-superimposable, right? This is my hand. This is my plane of mirror, and I fold it on top of each other. Is it superimposable? Is it completely looking like each other? Across a plane of mirror. Again, I will clear it out. Hopefully now, Priyabrata, this makes sense. When I said that enantiomers are mirror image, if I mistakenly said no, but non-superimposable, I will superimposable. I will clear it up right now. Hands on top of each other are non-superimposable. Hands across a mirror are exact images of each other. So they are mirror images. So mirror images cannot be superimposed on each other. So here across a plane, when you fold, these are mirror images. Mirror images, why will it not fold? Because when I take it away from a separate unit and put on top of each other, they will not superimpose. So those are called enantiomers. And just if you want to remember, just remember in terms of your hands. Hands are non-superimposable. Across mirror, they can sit on top of each other clearly. So that's mirror image. Enantiomers are non-superimposable. These are technical terms. Hands are the best example to understand them. I'm waiting for if anyone has any questions. I see someone typing something, so that's why I'm waiting for. Someone is typing something. Okay, I don't see the person typing anymore, and others are okay. I will come back to the chat box. If you still want to write something, please do. Right, now you have enantiomers, which are mirror images. You have, when across the plane, the images don't look similar. Those are called diastereomers. Now wait, let's 1 and 2 we have done. Look at third and fourth. CH3, CH3, X and Y, X and Y. X on the left side, for the other X is on the right side. For one Y is on the right side and one of the other Y is on the left side. So now if you fold it across a mirror, will they sit properly? Yes, because this X will come on top of each other, this Y will come on top of each other. So they are enantiomers. Okay. Ma'am, will we get the, yes, recorded sessions will be there. I think you've been given access to Excel sheet or a Google sheet for that matter, where you will have the link for the recorded sessions. It will be uploaded in YouTube. You will have the link for that. So you will get the recorded sessions for your producer later. All right. So mirror images are enantiomers. But now let's look into this first and let's look into third. CH3, CH3, X, H, Y, Y. Now, if you put it across a plane, they are no more looking exactly similar. Why? Because your X is on the same side. So when you fold it on top of each other, your X will go this side. While your Y may come and meet, but your X will not come and meet anymore. So such images, such kind of structures are called diastereomers. Enantiomers, opposite, diastereomers. You don't have to confuse yourself to a very greater degree. Non-superimposable, that concept is understood. Now, in non-superimposable structures, you have enantiomers, which are mirror images, diastereomers, which are non-mirror images. Example, if you remember one example of any, you should be remembering the other very easily. If you're doing it for the first time, it can look very overwhelming. If you are doing it after a lot of time, that also can be overwhelming. So it's okay. If you're confused, like I have just mentioned, that these recorded sessions will be available, make use of those resources. If you need book, please refer to what I have referred to for this class is Leninger. You can refer to Leninger. There are other books which also talk about these biochemical concepts, these stereochemistries. If after all of this also you have any doubt, there is something called discussion forum for the courses for NPTEL. You can post your questions there, or you can write to us if you want. We are always there to help you with this. Right. So you have enantiomers and diastereomers. And then moving ahead. So when you have the components that, like we mentioned, L amino acid, R and D amino acid. Similarly, you have some naming system which is called RS naming system. Now, when you are following this nomenclature, what do? We have an exercise on this, so it will be becoming clear to you. So this is the order of priority that you should remember. OCH2, OH, NH2, COH. Now, any question? Okay, so now you are given a compound and you are told to look into the compound and let us know what is the naming for this. Is it R or is it S? So how do you begin? You begin by looking at these functional groups here. Okay, in this example, fourth is the group with the lowest priority. And what groups are you looking? First, you need to find out your chiral atom. So chiral atom, as I told you, some atom which has 4 different groups. So this group has 1, 2, 3, 4 groups attached. Now, what do? You look into the least priority group. In this, which is the least priority, H. So let's say it was fourth was an H. So you start with this. Now what do? Now you do. So what you look into is what is the order or what is the priority of the other molecules. So 1, 2, 3 or whatever. You look into it. What you need to remember is how are you moving from the 1 to 3? 1, 2, 3, or how are you moving from the 3 to 1 direction? Okay. So if you let's move from 1 to 3. So after locating your fourth, keep your fourth at hold. Now you look into your other components, other functional groups, 1, 2, and 3, and see how are you moving from them. If you're moving 1, 2, 3 in a clockwise manner, it is R. If you're moving 1, 2, 3 in a counterclockwise manner, it is S. Just remember this. This would be the easiest way to remember it. Okay, we have an exercise. Just next slide. Let's do it there. It will become easier for you. Okay, but what do you do in RS naming system? You have a priority groups. In this, H is the least priority, OCH2 is the highest priority. You start by locating the least priority, giving numbers to the other groups based on their priority, and then move from that three priority group in their order 1, 2, 3, and look how are you moving, clockwise or counterclockwise. Okay, and then give respective name. To remember which one is what, a very simple thing that I do is, so clockwise is basically moving in this manner. When you are writing R, the first turn that you take is also in this manner. So you clockwise is R, and counterly, if you are moving counterclockwise, it's S. Okay. Let's move ahead. I post your questions in the chat box. I will definitely come and check it. Like I mentioned, stereochemistry is important. One, to visualize the structure. Second, the way it is spaced in the nature of it, the way it is occupying space is important for its biological role. A very prominent example is enzyme and its receptor, or cell surface receptor and its ligand. Their structure, the 3D geometry has to match perfectly for a ligand to dock with its receptor. So your stereochemistry tells you how is it arranged and lets you know how will it interact with its component, it's its interacting partner, right? Okay. Moving ahead. This is the example. This is the exercise that I was saying, but there is an exception which is known as vertical root method. Yes, science is full of exceptions. This is definitely so. What I have done is just keeping it very basic for you to give the how do I say, the basic principles there. But like Priyabrata has mentioned, there are always exceptions. You will go to internet, you will go to book, you will see that something doesn't follow the scheme. They are exceptions exist, and those are those exceptions. But when you are to understand the exceptions, you need to first look into the basics first. So follow with the basics, start with understanding the basics, and then you will, for you to grasp on the exception, which is very easy. Like Priyabhata mentioned, there is a method called vertical group method, which I am sure that when you go and look into it, you will see that they don't follow any of these norm, and then you have to do something else. But that's a different thing for uh, a bit more advanced biochemistry when you go into the next step. But for now, you can learn this basic naming system. One is RS, and we'll also do LNR, LND. Okay, so here is the alanine group. Let me know which configuration is this, R or S. You have H, you have COO, uh, R, S. Okay, people are writing their answers. S, okay. Yeah.
Ma'am, as
S, okay.
Ma'am, yes, ma'am.
Yes.
Okay. I guess I've received a lot of options and answers. Okay.
Yes, ma'am.
Yes. I am seeing most of you are getting it right. So I hope it was understood by everyone. Okay. Let me tell you why is it S. So you have H as the least priority. And then the remaining I have labeled as 1, 2, and 3 based on their priority. And 1, 2, and 3 I am moving in the counterclockwise. So it is S. I hope that is clear. I see 1 as R option. I hope this is clear to you. Why is it R? Not R, why is it S? Okay, let's move on. Now, these topics I did not discuss as theory, but they are also part of your assignments and different questions. So these are part of questions. We'll solve the question, we'll get to the theory. So you have two kind of projections here, Fisher projection and Haworth projection. Let me first take your guess on what do you think, which one is the correct answer for is, or false for that matter here.
Ma'am, second and fourth.
Second and fourth. Are you
Ma'am,
sure?
first
Second and
and
fourth, because they have the same projections. Just check once again.
first and third, ma'am.
First and third. Uh, you're all talking about the same projections in two forms. So Fisher is wrong for open chain. Fisher is also wrong for cyclic forms. I think you want to answer for Fisher and Haworth. So just check once again.
Second.
Okay. Okay. Let me come to the answer. So third and fourth. So yeah. On the left, the linear form, the open chain form is a Fisher projection. On the right, you have the Haworth projection. Haworth is basically you are just connecting this group and this group 2 and 5 and then making a cyclic projection. So the same structure, if it is in open chain formation, it is a Fisher projection. If it is in a cyclic formation, it is called Haworth projection.
Ma'am, the answer should be 1 and 2, no?
Fisher is typically used for open chain is a correct answer. The statement is, the question says statement is wrong. Fisher is used for cyclic form is a wrong statement.
Oh, sorry, I didn't see that what is asked.
My cue for the entire class has been look into the question properly. I have done those mistakes in my exams and I know how much it affects. Any exam that you take, GATE, NET, wherever, just be mindful of what you cross check and say those answers. And it's just healthy and advice. All right. It's okay. And don't worry about it. It's fine. It happens. Okay. Moving ahead. Now, let's come to DNL. But before okay, the DNN for some reason decided not to do the animation. I will ask you the question. So in living organism, you have amino acids and you have sugars. What form of amino acid and what form of sugars do you have? So you have levo and dextrose, right? Levodextrose, L and D. So do you have L amino acids or D amino acids in your living organism? And in sugars, do you
L,
have L sugars and D sugars? You can write it out. You can unmute yourself and say, for some reason, the
L,
yeah,
ma'am,
yeah,
L
you
amino and D
okay,
sugar.
okay. So L amino acid, D sugar. Anyone else with any other answer, or you all agree? Okay. I see answers. Yes. D, glucose, and L, sugars. Yeah. Now, let me tell you what is L and D in respect to sugar and in respect to amino acid. When do you call it L and when do you call it D for amino acid and sugars? Here on the bottom, you have sugar. When do you call um, glucose or a like a sugar as L or D? What you need to look is for the carbon atom, which is the farthest from the carbonyl group. Here the carbonyl group is here, CO, CO, CHO, CO. So the farthest C is this. Now for this C, you will look where is OH present. Again, the same principle as that of R. When you're writing D, your hand moves towards the right side, and OH present on the right side is D glucose. So if your OH is present on the right side, it's a D glucose. Opposite to it, if you have OH on the left side, it's L glucose. Right? Now, what do you look for amino acid where you don't have this kind of carbonyl and all this group? You look for amino group. If your amino group is to the left, it's an L. If your amino group is to the right, it's D. Again, same thing. Right side D, left side L. I hope this made it simpler how to name it L and D without forgetting the logic. If you're if it's a sugar, look at the farthest carbon from the carbonyl group. Look into your OH. If it's on the right, it's D. If it's on the left, it's L. Similarly, amino group for amino acids. If it is on the right amino group, it's on the right, it's D. If it's on the left, it's L. Did that make it simpler?
Ma'am, I have a doubt.
Yes.
Ma'am,
Yes.
is this L and D also for keto sugars?
Your voice is cracking. Do you want to type your question? It will make it easier.
Ma'am, is this L and D form are also found in keto sugar?
Uh, yes. I mean, we did discuss about glucose being a ketose sugar and aldose sugar in the first bit. So your L and D form are for sugars. So it will be there. Ketose and aldose is based on the group, the functional group that they harbor. But your D and L designation is based on this OH positioning. So it is also, you will see in an upcoming question where you will see D glucose 2 keto. So you have keto sugar and you also have these L and D. LD configuration. You also have on your Haworth projection something called alpha and beta. So you have multiple type of representation and naming of the molecule. Do you want to go ahead, Priya brother? Do you have another explanation? Or do you want to
Yes.
comment on this? Yeah,
Yes.
sure.
Yes. Yes. If I can explain in this way that if you see the bottommost chiral carbon, okay, and the hetero group means any other group that doesn't contain the carbon or hydrogen. So if it is in the right side, it will be D. If it is in the left side, it will be L.
Thank you, Priyabrata. Neha, did that answer your question?
Yes, ma'am.
Okay, thank you so much, both of you. Thank you. Okay, let's move to this, and this is what you see: D2 glucose. Okay, so this question is: you have two structures. These kind of questions will come up in biochemistries where you need to identify the structure or identify the compound based on the structure given. This is one of those questions. I have another such question. It's a good habit and good practice for you to look into different structure. So two structures are here and I have multiple groups on this options. Uh, let's, if you can try and attempting see which one is correct and see if you are understanding it. There is no logical explanation here. It is for you to understand the structure and know the structure very well, because these are very commonly used components, so compounds, and you should ideally know their structure.
Fourth option, ma'am.
Fourth option. Anyone else? Okay, people are typing. Let me just
E fructose and L arabinose.
Sec-
Rapinos.
Second option.
Second. Fourth. Second. Okay.
Third, ma'am.
Fourth, ma'am.
Keto glucose. Okay. Xylose. Anyone else? Two or four? Okay. I think I have received answers from most of you. Okay, it's two. Uh, just for someone who said third, keto, I'm expecting you have seen this group here and then wrote keto. Just remember when we were discussing about aldose sugar and keto sugar, we had this example there, fructose, and we had mentioned about this being a keto sugar. So that's why you get most often confused. But this is fructose, and the other one is arabinose. So, and the others which you have here on this slide, xylose looks like this. You can see how similar they look. Ribose, and this is D2 keto glucose. So this is the keto group here for the glucose. Okay. This is the other question. Again, all of them are D, of course, but what you need to know is which ones is the structure for what. Okay, someone else also written second that you got right.
Ma'am, second option.
Second option. Okay. Anyone else?
Ma'am, second, ma'am.
Okay.
Fourth, ma'am.
Okay. Okay. I will give the answer. So your A, this structure is T-glucose. I understand this is where the second came from probably, but this is your glucose. B, this one here is mannose. This structure here is erythrose, which is correct, which the smallest structure is erythrose. And the D here is galactose. So my suggestion, these structures of mannose, galactose, glucose, fructose, these you should definitely try and remember as correctly as possible, because these are the, see, we have done mannose and glucose for epimers. We have done galactose and glucose for epimers. We are now doing, we have done ribose, we have done erythrose. So these are some common and basic sugars that you should know that they often come up in different set of questions. Okay, I am left with very left time. So what I will do is I will try to cover as much as possible in the next 10 minutes. But whatever is remaining of this portion, I will continue this in the next session. It's a very important and a very interesting session, as you already have access to week 2's coursework. So you know that these third chapter, third lecture, and the next remaining weeks are all interconnected. So I will also continue in this in the next week. I have only few minutes remaining, like 10 minutes remaining. So let me just go over as much as I can. Right. So the last part for the first week was about water. And we have seen how water is a highly polar molecule, which is also capable of forming hydrogen bond with itself and of course other solutes. And it's a good solvent for polar solutes. And if it is polar solutes, it forms hydrogen bonds with it. And for charged molecule, also it interacts with it with electrons. So it interacts in both ways, forms hydrogen bonds with polar molecules and interacts electrostatically with charged solutes. Now, hydrogen or water molecule has this ionization potential. So capability, when you see H2O molecule, it can form this H plus and OH minus, a proton and a hydroxide ion, right? Now, hydrogen ions formed in water are immediately hydrated to form hydronium ions. So this H plus actually goes to the other molecule of H2O and this forms an H3O plus, this here. H3O plus and the OH is remaining. So there is this H plus and OH formation, but the H plus doesn't stay. It goes and forms the hydronium quickly with the other water molecules. And this leads to something which is also there in your lecture series, something called proton hopping. So you have this proton which keeps on hopping to the adjacent water molecule. So there is a hopping of proton as the next water molecule accepts those protons, right? Now, what happens? So this short hop of protons between a series of H bonded water molecules effect an extremely rapid net movement of proton over long. So you quickly are moving the H plus charge, right? And this is faster than true diffusion. And it shows the high ionic mobility of H plus. And this is why you have the polarity. And this is why you have solubility high in water, because the H plus moves very quickly. That moves because of this protein hopping, right? Now, this concept here, ionization over water, and the concept subsequently, which is pH, they're both interconnected to each other. So let's try to understand this. When ionization of water happens, how do the equilibrium constant look like? So on your left here, this is the basic, like the formula to look at any equilibrium constant. So if you have two reactants, A and B, and they give C and D product, Your equilibrium constant is basically looking into the product concentration divided by the reactant concentration. So CD and AB. Now we'll look into this formula when we look into ionization of water. So you have H2O gives rise to H plus and OH minus. If you look at equilibrium constant, you have H plus OH and H2 divided by H2O concentration, right? So this we don't know, but here what we know is in pure water at 25 degree, the concentration of water is 55.5 molar. This calculation is based on how much gram of H2O is there in 1 liter and which is divided by the molecular weight of water. So 1000 gram of 1 kg, which is divided by 18. So when you do this, you know that when you're looking at equilibrium constant at 25 degree for pure water, the concentration or amount of water that you have there is 55 mole. With this information in your hand, you can reconstitute this. You take 55 molar into K equilibrium constant, that becomes Kw. That is the ion product of water, right? Now, how do you find out what is Kw? What is this equilibrium constant actually? So what you do is you have H and OH. So you have 55 mole into 1 into 8 into 10, 1.8 into 10 to the power minus 16, which is this K equilibrium constant. Where is this coming from? We know that the value of equilibrium constant, which is determined by electrical conductivity experiments or measurements of pure water, is 1.8. So this value is also known. This value we have just derived. We now just need to calculate, multiply both of them, and we now know that at pure water, equilibrium constant is 1 into 10 to the power minus 14 m square. It is too much to understand, yes, but I will repeat it once again. Just let me walk through it once more. Now, when you know that this is the product of water, what happens at neutral pH? At neutral pH, you have H plus and OH is similar, is exactly same constant, because that's why it's neutral, both positive and negative charge being at the same amount. So you have Kw as, let's say, the square of H plus. So Kw becomes root square of H plus, right? Now we know Kw is 1 into 10 to the power minus 14. If you do square root of this, it is 10 to the power minus 7. So H and OH being 10 to the power minus 7. 7, pH 7. If you know the golden term 7, pH 7 is a neutral pH. That neutrality comes from here. Okay. I think so. This is what, okay, let me just do this much first, and then this is what translate into pH. You see 7 is neutral, and then as you go up, your OH minus concentration increases, and as you go lower, your H plus concentration increases, right? So your this ionization of water, this concept is related and correlated with your pH concept. I have questions, but I know that if I start that, we won't get enough time to do the numericals. So maybe I will let this be the last slide of today's discussion and just stop here and discuss more. So was this confusing, the last two slides? Or did you understand, or did you even get few parts of it to understand what we are trying to do here? Okay. Someone says clear. Is anybody with any slide of doubt? So on these two topics, I have at least 3 to 4 numericals. And numericals is what you have also in your assignment, I believe. So those numericals we'll do in the next session. But if you need any immediate help, I can show 1 or 2 questions. Electrical conductivity. Okay, just a second. I will just let you know. Anyone else? Yeah. So if you need any help with the numerical part of it, let me know. I can show 1 or 2 questions. If not, then we can just stay on this slide for the while. One into 10 to the power minus 16. Okay. Yeah. Let me see if I have the source for it that I can show you directly. Mhm. With me right now. Yeah, I have understood your question. Okay. Uh, all right. So Priyabrata asks me, can you explain the electrical conductivity? Ma'am, can you explain again about neutral pH? Okay. I'll take one question at a time. So the value of K could determine by electrical measurements of pure water is 1.8. If you want me to give the derivation of it, then I can explain it to you in the next step. Right now, I can only tell you what was the experiment that was done to, because for me to show you this, I need to go to other presentation. But I do have a slide on this. Maybe just let me check once. Do I? No, I don't. Yeah, right. I don't have it here. So if you want, do you want the derivation or have you understood? Are you confused with the term of electrical conductivity? If you want the derivation, I have it somewhere else. I have to show it in the next session. No for? Sorry, I didn't understand. I'm confused with so your value, right? The determination. So let me give it to you in the next session where I have those values with me and I can show you how I determine them. Will that be okay?
Okay, okay.
Sure, thank you. Coming to the next question, which says, could you explain about neutral pH? Yes, I will do that. So you understand till this part, right? I mean, the calculation that you saw about iron product. I hope this part, till this part was clear to you. 1 into 10 to the power minus 14. So what I said after that is, so if you look at this value, Kw, this is basically the product of H plus and O minus concentration, right? Now, neutral pH will basically mean that your H plus and OH minus are equal in concentration. So I have just substituted in place of OH, H plus, because OH plus and OH minus and H plus are basically the same concentration. And then I just found out the value for H plus. So if Kw, I already know my Kw, and I know H square is equal to Kw, I can find out my hydrogen ion concentration. Hydrogen ion concentration will be similar to hydroxide, which will be 10 to the power minus 7. Right? Now, if you look here, this is what I have to show you next. If you look at here, when you know the concentration of your H plus, you need to look at pH. Now, remember I told this is at neutral pH because I assume that H plus is equal in concentration to OH. When I did that, to find out pH, I just need to do log of or minus log of H plus or log of 1 plus H plus. When I do that, I get a value of 7. So that means at the pH 7 also translates to H plus and O minus being in the similar concentration. That makes the overall net pH as 7 or neutral. Yeah, I had also shown it here. It's been one of my questions. So, here. Yeah. Was the point about neutral pH clear? Okay, sure. I see Priyabrata has already written why he understood. Precisely that, 55.5 being the mole concentration. But I will still show it for in case anyone else had a doubt, and I will also put the other question that was put across to me as a reducing sugar, that also I will put in the next slide. So in the next slide, we'll begin with the route on reducing sugar. So please do join so that I can show you on that. And then I will also continue from ionization water once again, ionization, pH, and the next week's questions, and a lot of numericals on pH to help you understand the topic better. But we have to stop here because our time has exceeded. But in case anyone has any more doubt, I'm here. You can reach out to me via discussion forum, or you can mail me if you want, or let me know if you have any doubt. Or if you want any, if you have any inputs for me, how to increase the, or how to better the sessions, please let me know. Or do you think including numericals is fine? Or you want more of theory? Please do let me know. Because these are for your revision. So I would like it to be catered as to how you would rather need it. Be it when you are live attending live, or when you are attending or looking at the recorded lectures for your revision. This should help you revise and understand the topic better. So let me know if this is fine, or I should make any changes. And if there is any doubt which is still there, I will try my best to solve it. And if you have no doubt, then we are done for the day and we see each other next week. Thank you so much. Thank you. Thank you. Thank you.
Thank you, ma'am.
Thank you so much. Thank you. I'm sorry if I was a bit unclear. I'll definitely work hard in the next session.
Thank you, ma'am.
Thank you so much. Thank you.
Thank you, ma'am.
Thank you. Thank you so much. Thanks. Thank you.